Once we have collected all of the data from the fMRI scanner, we can begin the analysis. Analysis of brain imaging data involves numerous steps, including corrections for movement and transforming the data in a standard atlas data space. We have developed scripts that automate analyses of large groups of participants. Our data processing stream also involves a number of quality checks. It is very easy when analyzing brain imaging data to simply put one's data into the black box of the analysis and then only pay attention to the final product that is produced. However, numerous artifacts can hinder the proper interpretation of data that is treated this way. We prefer to look at the data at numerous points in the processing stream. For instance, here you can see an in-house program that we call a rainbow plot, which is a plot that shows the raw time course of BOLD, or blood oxygen level dependent signal from a region, with the different task conditions shown by different colors in the background. Here, the dotted time course is taken from a visual region in the brain, and we can see that the activity is lower during the rest periods, which are light blue. This allows us to check that the timing of the psychological paradigm, which we take from ePrime data output files, is properly aligned with the scanner data. The graph below the rainbow plot shows movement. This is a participant who moves a lot, and we can see that periods of high movement often correlate with noisy, bold signal. From time to time, we exclude individual runs or even whole participants when the noise due to movement is excessive. Once we confirm good data quality, we use FIDDLE, a Washington University St. Louis program, to create general linear models of the data for each participant. The GLMs allow us to make contrasts between each event and activity in the brain during each event. Once the contrasts are made, we can load them into CARET, another Washington University St. Louis program, to view changes in activity on the cortical surface. Here's the same image we showed you in the introduction, which compares social reasoning with scientific reasoning. In addition to looking at how functional data relates to task performance, we are also using a technique called Voxelbased morphometry to examine how differences in gray and white matter volume relate to individual ability in different domains. The functional data we have collected also affords more mathematically complex analyses, which are based on advanced mathematical methods that are currently under development to characterize patterns of connectivity as well as the complexity of activity within a region. This illustrates some of the principal points involved in a large and complex study, from data collection through analysis.